It is my pleasure to introduce Rachel Vannette. She's at the University of California, Davis. And I was writing up a little bio for Rachel, but then I was reading her lab website and I thought that she did an amazing job of explaining everything. So I thought I would just read about her lab. So the Vannette lab is a team of entomologists, microbiologists, chemical ecologists, and community ecologists trying to understand how microbial communities affect plants and insects. Sometimes we do other organisms too. We often study microbial communities in flowers, on insects, or in the soil. So with that, Rachel, welcome. It's so lovely to have you virtually and looking forward to hearing about your work.
Great. Well, thank you, Lauren. And I want to especially thank you for the invitation to speak with you all today. I'm very excited to share some of the recent work that we've been working on and get your feedback on some of the ideas that we're thinking about. So, in this new world of Zoom, feel free to put questions in the chat. I'll have it pulled up. So in case there's something relevant that, you'd like me to address partway through the seminar, I'm happy to do that, or, just raise a hand. I'm happy to take questions throughout. Just want to make sure things are clarified. So, so with that, I'll sort of dive into it. All right, so we in ecology and evolutionary biology, I feel like, are now recognizing that microbes affect species interactions in a number of ways. Those mechanisms have been highlighted previously in some really well-studied systems. So we know that microbes can influence host nutrition by accessing novel sources of nutrients, highlighted really well in pea aphids or legumes that associate with microbial communities that access novel forms of nitrogen. Microbes can also provide services like detoxification of diet. And that's a really good example of that is the Drosophila yeast system, where cactophilic Drosophila vector these yeasts along with them that help detoxify otherwise very toxic cacti that they feed on. Microbial communities are also well known to protect their hosts against pathogens or spoilage, really well characterized in this cool bee wolf system, where bacteria contained in the antennae of bee wolves and spread to their progeny help reduce pathogen growth and also maintain the quality of stored food for this organism. And we also are continuing to recognize the role of microbes in producing signaling molecules and helping playing an active role in communication among organisms. So today I'm going to sort of look at the role of microbial communities and how they might influence species interactions, but we're really going to focus in on flowers. And I think that flowers are a really important system to work in for a number of reasons. First of all, and obviously they sort of facilitate plant sexual reproduction through attracting pollinators and facilitating pollination, and then ultimately producing seeds and crops that we all sort of rely on for food. But not only the seeds themselves, but also products of the flowers, things like pollen and nectar, are key resources for pollinators, including organisms that we now recognize as really keystone species for ecosystem functioning, things like bee species or other pollinators. And so by studying the microorganisms that live in flowers, we might uncover some novel types of interactions that are essential for either seed production or pollinator health. And so previous work has uncovered at least a number of types or functional groups of microbes that we find within flowers. And perhaps one of the first groups that was identified as being important in flowers were plant pathogens. So many of these pathogens form visible symptoms on flowers, things like brown rot blossom blight, that's a major pathogen in almonds, or Erwinia amylivora, a pathogen that infects fruit trees, stone fruits, and can result in rapid limb dieback and eventually kill the whole tree. So flowers are pretty important infection sites for some of these plant pathogens. We also know that flowers are a sort of a hotspot of transmission for bee-associated microbes. So we'll find, or many folks have documented finding things like Nosema, Cryphidia, some of these bee pathogens on flowers. But not only pathogens, it seems like some other bee-associated taxa, things like Lactobacillus species, may also be acquired at flowers. And these microorganisms have been implicated in pollinator health. A final group of microbes that we find on flowers has, I would say, the least known about its ecology. And these groups of microbes, I'm going to refer to them as maybe commensals, maybe mutualists. We don't really know. All we know is we find them very often on flowers. And this might be things like yeasts or bacteria that seem to be quite specialized. We don't find them really outside of the floral habitat. So a little bit of background. Earlier work suggests that some of these yeasts that are found in flowers can metabolize nectar sugars and release volatile compounds that are detectable and attractive to bumblebees. And so it's thought that these bee-yeast interactions are pretty important to maintain yeast populations and sort of disperse yeast among flowers. Benefits for the bees, however, are really not well understood. And so this yeast bee interaction has only been documented in a few plant species. So when I got to Davis, I wanted to first examine how common are these yeasts among flowers? Are they found in many different species? We know that bacteria are there too. Who's more common and where do they occur? So in this talk, I'd like to focus mostly on some new work from our lab. And the first sort of general question that I'm going to address is what processes shape floral or nectar microbiomes, sort of at the landscape scale. And then secondly, we're going to zoom in and think about or explore the ecology of one of these groups of so-called commensal floral microbes. And then I'll talk about some of my future or current work that addresses some of these questions too. Okay, so the first part, let's think about the microbial landscape in flowers. If you walk out into a field, you might see a whole bunch of different flower species. What is the microbial landscape here? You might think about it if you're a microbe or if you're a bee. You might have variation in habitats. You might encounter different microbial communities depending on which flower you visit. So let's take a look. So one of the questions we wanted to ask here, do plant species vary in nectar bacterial or fungal presence and also abundance? Like where do we find these things and where are they abundant? And the second part of this, this question is what processes generate variation in microbial presence or abundance across the landscape? We might consider nectar chemistry, variation in dispersal, and you can probably think of some other processes that could generate this variation. So to take a first look at this, we decided to sample the microbial landscape by going to 2 different sites and sampling basically all of the flowering plant species that we could find that produce nectar. We also conducted this sampling across 2 different years. And in one of the sites, we had we started sampling the first year after a large fire went through. So these are also plant species that sort of pop up after fire. For each plant species that we were able to sample, we collected at least 8 and up to sometimes 200 flowers per plant species. Some of these plants have a long flowering period, some short, and overall sampled about 1,800 flowers to look at this study. And then we took the nectar samples and split them and plated some of the nectar on fungal specific media, some on bacterial specific media, and also validated that our cultures were either bacteria or fungi using a fingerprinting method called MALDI-TOF. We had confirmed that at least the large majority of our focal taxa were actually culturable using in a previous study where we compared sequence pools and culture-based methods. So we're pretty confident we're getting most of the things that grow in nectar. So what did we find? Well, if you go out and sample a flower in a field in California, in this part of California, you are about 50% of those flowers will contain some culturable microbes, and about 55% of them, let's say. And of those, most of those flowers will contain bacteria, and a relatively small proportion will contain only fungi. Some will contain fungi and bacteria. So you can see about 50% of the flowers contain bacteria, and about 20% contain fungi. So this is a bit lower than we were expecting for the fungi. If we break this up by plant species and look across many different plant species that we were able to sample with high replication, we find that plant species also vary in the proportion of flowers that actually contain microbes. So some plant species contain very low incidence of bacteria in the blue points and fungi in the orange points, whereas for other plant species, all of the flowers contain bacteria and many contain culturable fungi. So we've got a huge range here. We also found that plant species varied in microbial abundance. So here you can see in the blue points is average bacterial density in nectar, and we have average fungal density in nectar. And these plant species are in the same order as above. So a couple things we can conclude or take away a few patterns from these data. First of all, bacteria are more common and typically much more abundant than fungi in flowers. We also found that filamentous fungi are quite rare in flowers, and yeasts were really only common in these long corolla bumblebee visited species, sort of confirming what we had suspected, that the plant species that had been studied so far have been picked for specific reasons, or not all plants are similar to those plants that had been studied previously. The plants where yeasts were common were things like delphinium, cynoglossum, and castelliha. They have these long corollas. They're presumably visited by long-tongued species, including bumblebees. Okay, so bacteria are more common and abundant than fungi. We also found that plant species vary in the extent of microbial growth in nectar, often by 1 or 2 orders of magnitude. While that could be a temporal issue, we also suspect that some floral traits may enhance or reduce microbial growth. And so we wanted to explore a few mechanisms that may explain these patterns. So first of all, we wanted to look at dispersal and experimentally test, are bacteria better dispersers? Can this explain the pattern that we saw in the field of more common bacteria? And second, we wanted to also look at the potential for selection or environmental filtering. Can nectar chemistry alter microbial growth? Is it more difficult for fungi to establish in nectar than bacteria? Because that could be an alternative hypothesis for the pattern that we see. So for our first experiment, examining variation in microbial dispersal, a postdoc in the lab, Marshall McMunn, developed a kind of fun assay focusing on the Western flower thrips. This is Frankliniella occidentalis, a very common flower inhabitant. So if y'all have studied flowers, you've probably seen these, but you probably haven't quantified them. They are notoriously difficult to count, but they're often all, they love living in flowers. They eat pollen, they drink nectar, and they do sort of courtship behaviors on flowers. So they're very specialized to flowers, but they're also very prolificous. So they feed on many different plant species. So they could be a really good sort of model system to look at microbial dispersal among flowers. Ooh, they also attain really high abundance and are very frequently found in flowers, even though it's very rarely quantified. Okay, so we took these thrips from Diane Ullman's colony here at UC Davis, and we sterilized them by vortexing thrips in antimicrobial solution. They went for a little ride here in a pipette tip up and down, and then they got washed in sterile water. And we added them to a 96-well plate that had been previously spiked with a focal microbe. So these are, we're calling them sterile or sterilized thrips. And we allowed those thrips to feed and sort of go about their activity in the plate for 24 hours, after which we killed the thrips and allowed microbial growth for about 2 days. And then we monitored which wells, previously empty wells in the 96-well plate, were now colonized, and tested this across 6 bacterial strains and 6 fungal strains. So what we found was that, sort of consistent with our predictions, bacteria are much more readily dispersed by thrips among these sort of fake nectar habitats than our fungi. And we also had pretty low contamination in our controls. So you can see all of the bacterial strains were more likely to colonize new wells than were these yeasts. Okay, so this does seem to suggest that bacteria are better dispersers, at least via these really common flower visitors, thrips. We could also take a look at sort of this selection hypothesis. Do common components in nectar chemistry differentially affect fungi or bacteria, or different strains of them? And so to test this hypothesis, we also conducted a lab experiment where we took different nectar chemicals that are frequently found in floral nectar and have been sort of implicated in reducing microbial growth. Things like peroxide from Nicotiana, one of the floral volatiles frequently characterized from many different plant species, but certainly from penstemon, that's linalool. There are some alkaloids that are frequently found in nectar. We use delphiline, which has been isolated from delphinium. We also used a protein that has been implicated in reducing fungal growth, lipid transfer protein. And then we also tested ethanol, likely produced by microbes in nectar, and it may be sort of an antimicrobial product of microbial metabolism. And this work was done by a junior specialist in the lab, Tobias Mueller, and with some help by postdoc Jake Francis. So we took these compounds and put them again in a 96-well plate, added our fungi and bacteria, and looked to see how these microbes grew in the presence of these different compounds. And we found, so here's our scaled impact on maximum optical density. So it's basically comparing the microbial growth in the presence of a compound compared to its growth in the control, because many of these microbes varied quite substantially in the amount that they grew. So what we can find here, or what we see, is that peroxide is sort of a generally antimicrobial compound that we find in floral nectar, whereas some of these other putative antimicrobial compounds actually seem to promote microbial growth. This alkaloid contains nitrogen and may be accessible to some of these microbes that are found in nectar. Interestingly, we found no significant difference in the effect of any of these compounds on yeasts or fungi versus bacteria. However, we did find that, so we also included microbes that were frequently isolated from nectar and less frequently isolated from nectar. And we found that those that were rare in nectar were more negatively affected by these antimicrobial compounds. By some extent, not a huge extent, but it does suggest that these microbes that we frequently find in nectar are somewhat adapted to these chemicals. Okay, so what can we conclude from these set of studies? Well, we find, I think, fairly strong evidence for the assertion that both bacteria and fungi are dispersal limited among flowers, and that dispersal likely depends on vector identity. So we think that yeasts are probably vectored by large body pollinators like bumblebees, whereas bacteria are more likely dispersed by smaller organisms like thrips or potentially other flower visiting insects. We also find that nectar chemistry is variable, so we know it's variable among species, and we found evidence to suggest that that variation can affect microbial growth in nectar. And so this sort of leads us to conclude that both visitor communities and plant traits can determine microbial composition at sort of the landscape scale. And this was borne out in a recent study by Vannette, who quantified the microbial communities in a meadow in the Sierra Nevada, looking at both microbial community composition and very detailed measurements of visitor composition. And they found a relationship between visitor dissimilarity, between plant pairs, and microbial dissimilarity, suggesting that the composition of visitors to a given plant species does affect the microbial community. Okay, so hopefully I've convinced you that some of our normal ecological processes that we think about are also at play in shaping these floral nectar microbiomes. But from this, we also found, something sort of surprising. Yeasts aren't everywhere. And maybe perhaps they're not even the most important players in flowers. And so we really wanted to dig into some of the most abundant microbial taxa that we find here and try and figure out, what are they doing? These might also be really important ecological players. So for the second part of my talk, I really want to zoom in and talk about this nectar bacteria that we find to be very abundant, very common on flowers. And so this is called Tricks of a Nectar Bacterium, How to Crack the Pollen Shell. And the primary folks leading this project or helping with this project are a collaborator, Tori Hendry at Cornell, who has done a lot of the genomic analyses, Sean Christensen, a graduate student in the lab here, who has done a lot of the functional work, and Ivan Munkres, a previous junior specialist in the lab. So Acinetobacter, it is a bacterium. And when we have done sequencing of different floral communities, we find that it is very abundant, but primarily only after visitation by insects or pollinators. So here we looked at one plant species, Epilobium canum. We bagged some flowers to restrict pollinator movement, and then we allowed pollinator visitation, robber visitation, or pollinators and robbers to visit a flower. And you can see that this yellow bar pops up and is quite abundant in all of the visited treatments, most of the visited treatments. This is from the family Moraxolaceae, and is all of that family in this sequencing effort is comprised by the genus Acinetobacter. So we wanted to do some experiments with Acinetobacter, but for years and years and years, we couldn't get this bacteria to grow readily in the lab. So we decided, well, as our last resort, let's just sequence the thing. So we isolated a number of strains that wouldn't, we really struggled to get enough DNA to actually sequence, but we were able to get enough DNA. And as a result of these efforts, we actually described about 5 new species of Acinetobacter. This is the clade of flower-dwelling Acinetobacters. And you can see these outgroups out here contain mostly environmental Acinetobacters or human-associated pathogens, things like Acinetobacter baumannii, which is an emerging pathogen. So you can see these flower and also bee-associated Acinetobacters form their own clade. This is a picture of a bee here to remind me to tell you, really the only hit we had for some of these new species on GenBank was actually part of a Hapropoda bee genome. So it had been actually included in the genome assembly of this bee. So clearly these are, very pollination associated microbes. So if we zoom in just on this pollinator associated clade, we see we have new species called Acinetobacter pollinis, Barrettii, Rathii, and a couple others that aren't actually shown here. And through a genome comparison analysis, we wanted to explore what genes are actually found in this clade, and do they sort of differentiate the ecology of this group from the rest of the Acinetobacters. So in looking at the changes along this branch, we see that most of the genes that have been that differentiate this group are of unknown function, of course. Some are in energy metabolism, transport, signal transduction. So far, we didn't find anything that screamed, adaptation to nectar. Maybe some of the energy metabolism shifts may indicate specialization on certain types of nutrients. We also looked at different branches and different clades within this Acinetobacter group. And here we found that there were quite a few unique genes in this group that differentiated it from its relatives. And some of them caught our eye. So we saw biofilm attachment genes. Okay, that's really cool. But also cellulose and pectin degradation, in addition to phage and plasmid genes, sort of differentiated this group. And we noted that when we pulled those genes out and created a tree, there was a gene expansion in the pectin lyases and polygalacturonases. And so this is relevant to us because pectin is a component of plant cells, and it turns out of pollen cell walls. And so based on the previous literature that looked at bacterial strains or bacterial species that contain these genes, pectin degradation is present in a number of plant pathogens, things like Pectobacterium or Erwinia. These are known plant pathogens that can basically invade plant cells and degrade those using these enzymes. But honeybee symbionts are also known to contain these genes. And notably, none of these genes are present in the non-nectar dwelling Acinetobacter species, suggesting a novel acquisition of this sort of gene repertoire. And so we took that and sort of hypothesized that perhaps these genes might underlie interaction between the bacteria and pollen. And interestingly enough, pollen is pretty tough, right? So pollen is comprised of many different layers. So we have the exene, which is the outer sort of layer of recalcitrant material. Within that, there's an additional layer, this intine, that's sort of a protective layer that's comprised of pectin and cellulose. And so to get at sort of the nutrient-rich interior of the pollen, the microorganism or organism that's consuming the pollen has to get through the exene and the intine. So we wondered, how does it do this? So to start addressing this question, Sean collected pollen from the California poppy, Eschscholzia californica, and put it in a media that was intended to germinate that pollen as best we could. And this is a media that's typically used to assess pollen viability, especially for agricultural purposes. So in the control condition with no microbes, you can see pollen looks like pollen at 15 minutes, and at 45 minutes, still kind of looks like pollen. So that's cool. It's not germinating quite yet. But when they added the bacterial taxa to the pollen, at 15 minutes, the pollen, you can start to see, starts to germinate. And you can see these sort of little pollen tubes bursting out. And at 45 minutes, you can see the pollen tubes are actually much longer. And eventually, some of these pollen grains start to burst or the pollen tubes actually start to burst. And so here, there are some photos of what these different states look like. Here's the initial pollen, germinating pollen, burst pollen, and pollen that has germinated and then burst. And so to assess if this was simply, the one strain that we picked, or if many microbes also contain the ability to affect pollen in this way, Sean chose a number of different Acinetobacter species, Acinetobacter apis, Poseri, nectaris, and the new species Pollinus. So all of these are from that nectar-associated clade. And we also compared their effect on pollen to that of Pectobacterium, which we know contains a lot of pectinase genes, which we think were responsible for this phenotype, as well as the nectar yeast, Microbia ricofii, and a control. And so what we wanted to see is, over time, do these different microbes influence pollen germination? And so here we've got a 90-minute timeline. And what you can see is the control is down here. About 8% of the pollen grains germinated in that time frame, whereas and the Pectobacterium and Microbia yeast are also down there and not distinguishable from the control. As in comparison, all of the Acinetobacter species cause this pollen to germinate and start to grow a pollen tube. If we look at the fate of that pollen, we see that most of the pollen that has germinated also bursts later on. And we don't expect this to be 100% because some pollen grains are aborted or not viable or things like that. So, okay, so we've seen that that Acinetobacter can induce this sort of pollen germination and bursting, but then we wanted to see, well, do they actually benefit from this? And so to examine that, we took pollen and either kept it live or able to germinate, or microwaved it so that it was unable to germinate but didn't change its nutrient composition. And we compared microbial growth in both of these conditions, along with a no pollen condition, and simply the pollen by itself, either left viable or sterilized ungerminable. And we compared microbial density of two different microbes. One is Acinetobacter pollinus, and the other one is the nectar yeast, Microbia ricofii. And we looked at their cell density at 0 and 24 hours. So the next two plots that I'll present are Acinetobacter on the left, Microbia on the right. And so you can see that alone, we had fairly low growth from 0 to 24 hours for Acinetobacter and for yeasts. And that pollen by itself had relatively low levels of contamination. When we added the pollen to either of these, we saw both the bacteria and the yeast increase their growth quite dramatically, but only Acinetobacter benefits from the addition of this live pollen, suggesting that Microbia the yeast can use pollen nutrients, but doesn't seem to benefit from any of that pollen physiology or germination, which we showed it didn't induce germination. So that's probably a function of that. Okay, so Acinetobacter seems to induce germination, also benefits from it. But what led us to this in the first place was this potential effect of pectinases. And so far, we saw that Pectobacterium had no effect on pollen germination. So we wanted to do one more assay to see, well, do these strains actually produce any detectable pectinases? So we used a plate assay where you impregnate agar plate with polygalacturonase and grow microbial strains. In this case, we have 3 microbial spots here, and a clearing zone indicates degradation of that polygalacturonase by microbial enzymatic activity. And what we found is that our positive control worked great. Pectobacterium can degrade that material, but none of the other strains that we measured could really do the same. You see a slight clearing around Acinetobacter pollinus, indicating perhaps some activity, but this was inconsistent and also much reduced compared to the Pectobacterium. So it may be that the pectinase enzymes that are produced by Acinetobacter are different in their target site or active sites than those of Pectobacterium. But basically we're left with a little bit unclear answer on whether or not pectinases are involved in this process. So what can we conclude from this study? Well, we found evidence to support that that Acinetobacter does influence sort of the physiology of pollen, and in this case can trick it into germinating. And this may be a promising system to study the mechanisms involved in pollination or pollen germination, which is not well studied in many species. Let's see, so here we also have conflicting evidence for the role of pectinases in this process. Despite the diversification in this pectin lyase gene family, we didn't find strong evidence to support that it was involved in this process, but our work on this is ongoing. We also haven't yet explored whether this effect is species specific or more general to many different types of pollen. We've done a few studies that indicate it's likely to be more general. So this is a species of Clarkia pollen with no bacteria or with Acinetobacter, suggesting that Acinetobacter can localize around protein or around pollen and potentially do something to that pollen. We also are quite interested in the consequences of this microbial effect on pollen for plants and pollinators, or yes, plants and pollination. So for most plant species, pollen that gets into nectar is probably lost from the plant reproductive sort of track, except for in some plant species. So for example, Asclepias syriaca receives a pollinia or a packet of pollen that germinates in its nectar and then goes on to fertilize the plant. So in some cases, microbes in nectar may actually affect plant reproduction through this route. One other potential consequence of this that we're quite interested in is pollen eaters. So we know that many organisms consume pollen, and the mechanisms of pollen digestion are not well understood. So this is the hypotheses from Ty Rolston and Jim Kean's paper for hypotheses on how organisms acquire pollen nutrition. Some organisms crack the pollen wall mechanically, some pierce it with their sharp mouth parts like thrips. Some are known to dissolve pollen walls with enzymes. These first three are quite rare though, and really only described in things like Calymbula or thrips. But much more commonly, it's thought that bees and other organisms may induce sort of a pseudo-germination to happen in the gut, or burst the pollen wall with an osmotic shock. But it's difficult to distinguish these things, and not much mechanistic work has gone into it. And so we sort of compare the effect of Acinetobacter here to what has been documented in some of these organisms previously, just simply by dissecting their guts and looking at pollen morphology as it moves through the digestive tract. So we certainly see some parallels there. And so this makes us wonder, is there a role for microbes in pollen digestion? Perhaps there's simply convergent mechanisms that organisms use to acquire nutrients from pollen. But we're hoping to look into some of these possibilities. And so one of our current directions is trying to understand if these floral microbes are obviously consumed by many pollinators at flowers and obtained at flowers. And they're thought to be quite important for solitary bees and comprise a large proportion of their microbiome. And so we are interested to find out if some of these floral microbes can affect provision quality and bee health. And so we're looking at this in a number of species, including Osmia, and also this cool bee that nests at the beach, Anthoptera bomboides. So with that, wanted to wrap up, give some time for questions, and also acknowledge the many folks that have collaborated on this work, and also my funding sources. So thanks for your attention, and let me know if you have questions or ideas. Yeah, Matt.
Hi, Rachel. Can you hear me?
I can hear you.
Cool. That was great. Thanks a lot. I have a bunch of questions, but maybe I'll ask one or two, and then if there's students who want to jump in after me.
Sure.
So you mentioned initially that the Acinetobacter you have weren't growing super well. It sounded like you got them at the end for some of the experiments you mentioned, but all this was pointing towards, had you tried like supplementing your media with pectin? I just was wondering if like that was sufficient to get them over the hump.
Yeah, that's a great question. So we tried this polygalacturones, and they don't grow very well on it. If we add pollen to the media, they grow great.
So there's something in there that they like.
It's something in there. Yeah, exactly. So we've been trying to get different fractions of pollen and try and figure out what exactly they're using. We haven't quite figured that out yet, but it seems like they like some sort of the protein or some component of the pollen. Yeah.
Cool. And then from the early part of your talk, I think there's a lot of sort of ecological questions to ask jumping off of that. But one of them, the first one that came to mind was like priority effects with the flowers, right? So do you have any indication whether, the bacteria are probably better dispersers, but if certain taxa get in there first, are they inhibiting the yeast or other bacterial, it obviously gets complicated fast, but.
Yeah, definitely. So we've done during my postdoc, I did some work on priority effects with Tad Fukami in this system. And so we have a pretty good idea that priority effects are quite important they could be quite important in flower microbiomes. So I would guess that yes, they're probably very important. In some of the lab experiments we've done though, we haven't seen strong competitive interactions between, for example, the yeast and this Acinetobacter species, suggesting that maybe there's something else going on, potentially releasing nutrient, or they may be sort of specialized on different nutrient sources and able to partition things. So I feel like not a lot has been done in sort of real flowers to get at that question.
Got it. Thanks.
Yeah.
People are also welcome to type in the chat if they have any questions.
I'll ask a quick question, Rachel. That was
Sure.
Great. Thank you for that. Just to clarify, I'm not sure I understood how were you characterizing communities on the whole flower or only in the nectar?
Yeah, for most of the studies I talked about, it was just nectar. For the last one, it was whole flower communities. So we've done both. Yeah.
So I was thinking through like the implication for the plants in the pollen tube germination. So if it germinates on a stigma, for example, are those bacteria ever found on the stigma? You mentioned one case where something like that happens, but.
Yeah. Yeah, we have also wondered the same thing. So last year we went out and sampled stigmas, and yes, Acinetobacter are found there. We need to do a little bit more work to figure out if they're metabolically active and if they can affect that process actually on the stigma, because there it may be actually more important for plant reproduction, for example. Yeah, great question.
That's the other, I guess, part of it I was wondering is the bursting of the pollen tubes. I'm not super familiar with, but does that happens on stigma as well?
Yeah, so it's actually part of the pollen developmental program. So pollen, lands on a stigma and then undergoes some sort of hydration and grows the pollen tube down through the, germinates on the style, grows or germinates on the stigma, grows through the style, and eventually when it reaches the ovary, does that sort of bursting. And so it seems like maybe it's sort of bursting prematurely, or the Acinetobacter can sort of trigger that, that mechanism much earlier. Yeah. For flowers with low microbial content, are the microbes more likely to be pathogens as compared to microbes found on flowers with lots of microbes? That's an interesting question, and I don't know. For what we have found, I would say simply looking in nectar, Acinetobacter and Microbia are, if they arrive there, they are much likely to grow to very high abundance. Some of these pathogens can sort of hang out in nectar or on surfaces, but they need to invade the plant to grow to relatively high abundance. And so I think they have sort of different ecologies or strategies on the flower that you might see play out in slightly different ways. I hope that answers the question. Yeah, Peter.
Do you know, does anybody know if the, when the bees store pollen, if you know what's growing on it, what bacteria are in it when they do that? Like,
on the pollen itself?
Yeah, I mean, as indication whether the bacteria might be important for helping the insects digest it or get into it.
Oh, I think I understand the question, but I'm not totally sure. So how can we understand if some of these microbes could be involved in insect digestion of pollen?
Yeah, I mean, I was thinking about particularly the species that store pollen.
Oh, yeah. Yeah, so it seems like adult bees have mechanisms to access pollen nutrients. So they have a crop and they have a gut. And so they can, sort of pass pollen from one compartment to the next. And both those compartments differ quite a bit in osmolarity. And so they seem to be able to burst pollen. On the other hand, other types of pollinators don't necessarily have that sort of gut morphology. And so they may have to rely on other mechanisms to access that pollen nutrition. And so, yeah, I think trying to understand what microbes are there and how, if they're involved in that process, is sort of our next step. So I don't know. Yeah, maybe I missed something there. So let me know if I did.
Well, Rachel, thanks so much again. We have a kind of coffee or tea hour or tea chat time at 1:15, but we'd love to give you a 10-minute break if people are done with questions. Oh yeah.
Oh, one more, sure.
Okay.
Thank you for speaking. I was wondering how the proportion of yeast that you found in nectar compared to the proportion of yeast species in the general environment, because there were less yeasts than bacteria found in the nectar, and I was wondering if that was comparable to the overall environment.
Can somebody repeat it? It was a little difficult to hear.
Yeah, do you guys have a microphone? Yeah. Very echoey.
The mic's being weird today, sorry.
Is that any better?
That's way better. Yeah.
Okay, cool. I was wondering how the proportion of yeasts compared in nectar to the proportion of yeast in the overall environment surrounding the plants that you sampled.
Oh, great. So which kinds of environments are you thinking? Sort of like the plant itself, like the rest of its tissues, or other things?
Soil, actually. Yeah.
Oh, yeah. That's an interesting question. We don't know because I guess we haven't really looked. But the the yeast that we find in soil, I mean, they're certainly there, or different yeast species are certainly found in the soil, but we don't typically find these species like Microbiomes or Ricofii or bee-associated yeast. We don't find them in the soil. So it seems like they are really associated with some of these pollinators.
Are you seeing generally like lower quantities of yeast in soil and nectar? Are they just rarer than bacteria across the board?
Yeah, we haven't really looked. It seems like the flower is a very specialized sort of environment that may select for yeast, which like really sugary sort of this kind of environment. And so we think that their primary role is there. I mean, it certainly may be that there are other yeast species that are traveling, from the soil to other plant parts, but we haven't seen that. And I don't know if any other groups have really reported that either. Yeah.
Cool, thank you.
Yeah, thank you.
Shall we thank Rachel again?
Thanks everybody for your ideas and questions.